change this amino acid sequence, what's going to happen? So you had to know amino acids come together to make polypeptides. Polypeptides, thank you, go together to make our proteins, and then it's a change in the amino acid means it's a change in the R group, and a change in the R group means that we change how it bonds. And if we change how it bonds, we change the shape. If we change the shape, we change the function. And you see how you've got to know all these tiny details, but what's more important is understanding how everything affects itself, right? So as you're studying, Chapter 6 is all the organelles and how they work and stuff like that. But think about how even just yesterday, we were looking at the endomembrane system and talking about the fact that we go from ER, where it's made, the Golgi, where it's packaged and processed, move through the Golgi, where then it's sent out to the cell. Once it's sent out to the cell, it can bind with the cell wall or be kicked out of the cell completely. But now, not only do you have to know the little tiny details of what does each thing do, but how does that flow work? How do they go together? That's going to be important in order to do well in this test. So as you're studying this weekend, start with the details, but then go bigger, broader picture. Why does this matter? What does it affect? How does it relate to other things we've talked about? Does that make sense? Cool. So I wanted to go ahead and put that out there.
Okay, the question's about that thing, so it's like a T. So is it just like the cell losing and gaining water? Just how
Yes.
it loses and gains water?
Yes.
Okay.
All right. And so first, we're going to look at the cells without cell walls and how are they affected. So what would these cells be called? Animal cells. Yeah. And then we're going to look at cells with a cell wall, so our plant and our bacterium. All right. So if we, and now here's the other thing I want to clarify. When we talk about the tonicity of something, so if something is hypertonic, isotonic, or iso or hypotonic, we need to define, are we talking about solution or cell? These three things here are referring to the solution. Because what do we say hyper means? More or excess. Iso, same. Hypo, less or not enough. So if we're saying that this farthest to the left is hypertonic. Hyper means the water is going to go towards that thing, right? Well, where did the water move in that picture? Out of the cell. It would only move out of the cell if that was a hypertonic solution. So it's all about defining what is hypo, iso, or hypertonic. Is it the solution or the cell? And I know that threw some of you on that quiz, right? That you were like, is it the cell? Is it the solution? I don't know. It's a careful reading thing. Or if you're describing it, it's making sure that you are describing it correctly. So what we're looking at is a hypertonic solution, meaning that there are more solutes outside the cell than inside, which means that the water rushes out of the cell. If you had to describe that cell, what would you describe the cell the farthest left? Farthest to the left. The solution is hypertonic to the cell. So the cell is hypotonic to the solution. You see how it's all in phrasing? So it's a comparison of where the solute concentration is, right? So let's say that I have 5 solutes inside the cell and 10 solutes outside. So we know there's a greater concentration outside than inside, right? So I could phrase that 2 ways. I could say that the solution is hypertonic to the cell, meaning it has more than the cell does. Or I could phrase it that the cell is hypotonic to the solution. And again, it's because the cell has less than the solution. So it's all about which one are you comparing to which. All right? Yes, sir?
So the pictures, when it says like what type of tonic it is.
It's the solution.
We're talking about the solution and
Correct.
not the cell.
Yes, sir. Yes, ma'am.
So if the solution is hypotonic, then the cell is hypertonic.
Yes. Yeah, and that's the other thing. The only way they could be the same is in ISO because that's the definition of ISO. You're struggling with that thing over there.
I know.
That was a weird sneeze. You had like a whole body convulsion. Well, bless you. All right. So, yes, ma'am.
Do we need to know like what kind of molecules can like just randomly go through the membrane without a protein, and then like what goes passively with and what goes actively?
Your generalizations, you mean?
I don't know, like what kind of molecules, like.
So, like things that easily pass through the membrane without anything are going to be our hydrophobic or non-polar molecules. And that was in one of our first slides, right?
Oh, okay.
Everything else needs some sort of a protein. Now, active versus passive, there is a difference. Small polar, so things like water, oxygen, carbon dioxide, they pass through easily, therefore passive. But as we get larger or more charged, things like that, it takes more energy. That should be somewhere in there in your book as a, I'm sure there's like a one sentence. I would know the generalizations, yes, but like we're not gonna get into the details of active, and there's a whole bunch of different ways active. Don't worry about which one goes which there.
What happens to the cell if it's hypertonic, etc.?
If the cell is,
Yeah, if the cell is hypertonic, what happens?
so if the cell is hypertonic, that means water is going to rush in and swell or burst it.
So, but the solution's hypotonic.
So the solution would be hypo, yes.
So it's sending water in to try and balance.
Yes, ma'am. All right, so we're gonna, I know it's gonna seem redundant, but we're showing each of them separately right now.
Do all solutions end up being isotonic?
They're all aiming, wow, I lied, in animal cells.
Okay.
We are aiming for an isotonic solution or environment.
So eventually we get there, right?
Not necessarily, no, because they have to have the water available, or think about it with the blood cells. If we have so much concentrate of solute inside, we rush too much water in, the cells burst, right?
Yeah.
So if it happens too fast, so we move too much water, or we can't, because water's what's going to move first, easiest, most. So it's rushing in. We can move the solute out, it just takes more work. So if the water keeps rushing in to try and flush it, it could actually end up bursting the cell and therefore never reach it. Ideally, yes, we're trying to get there. Doesn't always happen.
Wait, so just water moving in and out, right? It's not like anything specific, right?
No, not technically. We are going to be trying to move some of the solute, but water's the one that moves easiest, moves most, moves quickest. So that's what's going to try and happen first.
So what's actually moving in and out of the cell to make it hypo and hyper?
Technically both, but the majority of it is the water.
But we just need no water.
Correct.
Okay.
All right. So with ISO, there's no net movement. Basically, it's moving in and out at the same time. We talked about that, right? Hyper, solution of solute, and this is talking about solution. So the concentration of solutes is greater inside than, the concentration of the solution is greater than inside. So the cell is going to lose water. The process of losing water in a cell is called plasmolysis.
Like 10 water molecule, or 1 water molecule goes in and like 10 would leave? Or like, that's like the retrospect.
For hyper,
Yeah.
it would mean, right, that if 10 leave, 1 might be going in, but net is still a leaving.
Yeah.
So leaving wins. Yep. And so for hypo, it's the opposite. And so the cell ends up gaining water.
Would they want to be like too full because it has a lot of water and solutes?
We'll just
Say that again.
This one cell bursts.
Like, wouldn't it end up being too full because it has a
It'll
lot of
burst.
solute and a lot of water?
If it's that much of a difference. Because remember, it's still a comparison, right? So like if we had nothing outside and 100 inside, then yeah, it's probably gonna burst because we rush so much water in. The cell is also at that point going to try and pump out some of that solute. That just takes more effort. So it takes longer, and that's why we don't typically consider that happening. So yeah, it can be dangerous for the cell if that difference in concentration outside and inside is too different.
So during like things such as like endocytosis and like exocytosis, does water ever go out through the cell through that? Like, through endo-
Yes, but in small amounts.
Okay, so it doesn't affect like the hypotonic solutions.
Yeah, because remember from the video there was a specific type called penocytosis, which
Yeah.
is the movement of water through that. That's no longer osmosis happening, so it just changes the whole process a little bit, right? So when we talk about animal cells, then so this is what you're looking at. If we have a hypotonic solution and it's too drastic, the cell will actually lyse, which means to burst or to explode. An isotonic solution would be normal. That's what we want in an animal cell. And if we have a hypertonic solution, it's going to shrivel up.
So all of the cells in our body are isotonic.
Aiming for isolate at least. So when you become dehydrated, that changes it. That puts you in a slightly hypertonic solution.
And what effect does that have on your body if you're like more hypertonic or like your cells are more
You get
hypertonic?
dehydrated.
I was going to say it affects whether you're dehydrated or overhydrated.
Okay.
Can you be overhydrated?
Our bodies are pretty good at processing it, so you pee more. But yes.
So hypotonic would be more.
A hypotonic solution
Solution would be
mean more inside than outside.
okay, and that's more hydrated. Like you would be overhydrated if you are okay.
So if there's a bunch of excess water, just like filters down, like sends it away. Does it want it?
As long as your kidneys are functioning correctly. Not, we got other issues.
Seeing the word thought. I like the only word I was putting on.
It's an important word.
What do you put? Doesn't water go from higher density to lower density?
No.
Higher concentration to lower concentration?
That's when we talk about solutes. So solutes move down their concentration gradient going from high to low.
Solutions don't change.
But that's talking about solute concentration. When we talk about water concentration, we're literally trying to water things down.
Oh, it says like the opposite. Oh. That makes sense.
You got it?
Wait, so it's water versus water in this, so it's like water inside concentration, water outside concentration.
No. It's concentration of solute. And the water moves trying to literally water it down.
Okay, that makes a lot more sense.
Yeah.
What? I'm overthinking.
So it's like
it. I'm overthinking it, yeah.
Yeah.
You always do.
Most of you do. Alright. So now when we talk about cell walls, basically they help to maintain this balance of water. Right? I love how like y'all each bring someone new. So when we talk about how the water balance is being maintained, it affects what we call turgor pressure, which is just the pressure of the water inside pushing out on the cell membrane. It creates a firm, tight state of this membrane, and that's a healthy state for plant cells. So we actually want a hypotonic environment in plant cells because that pushes just a little bit more water into the cell, and as it pushes a little bit more water into the cell, it makes it a firm, sturdy plant cell, bless you, and that's what allows the plants to hold up and stand up on their own. If the plant is considered flaccid, or wilting essentially, it's because it would be in an iso or a hypertonic solution. We're going to see pictures in a second that kind of show this, but this is part of why a second ago I clarified with Elizabeth's question was what environment do we want? It depends, are we talking plant or animal? In an animal we want an isotonic environment. In a plant we want a hypotonic environment, but it's because of the cell wall. So you know how I was telling you big picture? Here's a good example of that. Is I could tell you I take plant cell and an animal cell and I stick them in a hypotonic environment. Explain what happens. First of all, you'd have to know the definition. You'd have to know what is a plant cell versus an animal cell, what does hypertonic mean, or what does hypotonic mean. But then, bigger picture than that, you would have to understand how it affects the two differently and why. And going into the fact that an animal cell does not have a cell wall, only a membrane, and that the membrane is able to undergo osmosis, which means the water moves easily in and out of it. The plant cell, on the other hand, has a cell wall which holds on to the water a lot easier, and so it's a lot more difficult for things to move in and out, and being able to explain the whole thing takes a little bit more effort and knowledge than just straight definition type stuff. Right. So, yes, ma'am.
So if it's flaccid, like it's saying isotonic or hypertonic environment, that means the solution outside, right?
Yes.
Okay.
Yes, sir.
Do like willows not want to be turgid or like are less hypotonic because they like
It means the
wilt?
cells don't function.
No, it's not a true wilt.
Okay.
That is because of other things in those cells. That's just how they
I think
grow.
in terms of the cell.
Think of it more like once you have like cut flowers, they can take up the water for a certain amount of time, but then the phloem and the xylem start to shut off and seal off, and they can't take water up anymore, and it literally starts to become weak and go like that, right? That's because of that happening.
So they want to be in a more hypotonic environment.
Correct. For plant cells.
So the cell should be more concentrated with the solute.
Yes.
Okay.
Alright. Everyone got this stuff? We can go into the slide with the picture and explain it?
Yeah.
Okay. So here's your happy little picture. So if we look all the way up to the left, that would be a hypotonic environment. There's more solute inside than outside, which rushes the water in, and it makes the cell kind of just bulge a little bit. Well, that creates this pressure on the cell wall that pushes on the other cells, and it allows it to stand rigid. When it's just a flaccid or a isotonic solution, it's still going to create the structure of the cell wall, but they just fit perfectly in there. There's nothing keeping it snug. And then if we had a hypertonic environment, what happens? Well, the cell wall is relatively rigid, but the cell membrane inside isn't, and so it starts to pull in and away from the cell wall, and it starts to deform it even just a little bit, but it's going to shrink up how much movable space we have. So it's like it's shriveling, but the wall still stays intact. I would try and draw some sort of a little picture.
And it's different with plant cells because they want isotonic and that's where they're normal. Animal cells, sorry, animal cells.
Animal cells,
Yeah.
correct, yes. doing to him?
Oh my god. Tickles.
Your pencil fell on his back.
Oh, I was doing this. Yeah, I fell.
Okay. Let's look over and you're reaching down the back of his shirt. I didn't know if you were doing like what I did the other day. It's like your tag's hanging out, but usually you're like just at the top of the shirt for that, and you usually let them know, hey, I'm fixing your tag. And they're like, ah, why are you touching me? You know.
He's okay with that.
But it was a little strange. Not gonna lie. I look over.
I always do that.
Then again, you're freshmen. I've learned nothing is normal. So.
Well, I mean,
What?
I mean, let's go back to the beginning of class. Nothing is normal. So.
Well,
That's pretty much the one
the
main
beginning.
thing. That's the one main difference between freshmen and everybody else.
Yeah.
Actually, everything is normal.
Freshmen trip on backpacks.
No.
Oh no, I trip on everything.
Alright.
I trip on my
I
So-
tried to orchestra concert when the camera was on.
I'm sorry.
I'm sad.
Question for you. If I have 100 milliliters of glucose in solution A on one side of the membrane, and I have solution B on the other side with 100 molecules or milliliters of fructose, what's the water going to do?
Oh. Go into fructose.
Both ways.
Alright, so we're going to get everyone to weigh in. You have one of three choices. You can either say the water is going to move from solution A to solution B. You can say the water is going to move from solution B to solution A. Or your third choice would be the water is going to move between A and B equally. So everyone has to pick one. You gotta raise your hand for this. Who believes that the water is going to go from solution A to B? Own it. Even if you're wrong, own it.
Or that the water is?
Oh,
The water. We're talking about water. Alright? Who believes that the water is going to go from B to A?
Right here, guys.
Everyone had to say something.
Which one are
There's
you voting
third
for?
choice.
Ow, my shoulder.
Who believes that the water is going to move between A and B equally? Okay? Of the big group who wants to explain themselves. Alright? Yes, ma'am.
So, I guess the solutions are both, well, I guess since they're both equal, they're isotonic, and then the water will go in and out equally.
Perfect. Alright?
Dang it.
So, both solutions have an equal number of solute. Does it really matter what the solute is in this scenario? No. So they have equal amount of solute, so they would be isotonic to each other. So the movement is going to be equal between the two. Alright? Good. How about this one? Solution A has 100 molecules of glucose. Solution B has 75 of fructose. Alright? Again, I want everyone to try and vote. So who believes the water is going to move from solution A to solution B? Who believes that the water is going to move from B to A?
Right here.
Who believes it's going to move equally?
No, you're here.
Well,
Alright, from the group that said B to A, who would like to explain themselves? Olivia?
Okay, I got this. So if it has more molecules of glucose, then that means more solute, and so water goes in to try to like drown out the solute.
So we're going to move
Yeah.
the water from B to A because the solute is more concentrated in A and we're trying to water it down. So it goes from B to A. Alright, if you missed it, make sure you get this written down.
I don't get it.
Alright?
Yeah, same.
Okay.
Like, I understand what you're saying, I just don't understand how it works.
What do you mean?
Just saying how it works.
Osmosis.
No, I mean, like, there's 55 molecules, or, there's 75 molecules, and there's 100 molecules. Why would one with lesser molecules go to the bigger one? Like,
The
what is-
water, not the molecules.
Oh, okay, I get it now.
Okay.
Because there's more water in one.
Correct.
So it's trying to get
Because it's basically, when we talk about which way are things moving, it's a concentration thing, which is basically talking about ratios. So we're saying we have an equal amount of water, because we're saying per milliliter. So we have more molecules per milliliter in A than in B, which means A is more concentrated than B. So the water is going to go from B to A, trying to equal it out. Alright, yes ma'am.
Where do you get the water from?
It's dissolved in it.
Oh, wait, so does
Okay, so if it's dissolved in it, then wouldn't A have more water? Because it has.
No.
wait.
Hold on.
It's like one.
Think of it this way.
Water on each side, and one has 100.
Hold on. I'm gonna answer this and then this might answer other questions. And if not, we'll keep going.
It's without a development.
Alright, so. Alright,
Stop.
Ireland.
Stop, please.
alright, so what is that
Just let it
represent?
film.
The membrane.
Semi-permeable.
It's a
A
membrane.
semi-permeable membrane, okay? Blue dots are gonna represent water, okay? So I've got 1, 2, 3, 4, 5, 6. And I'm gonna put equal numbers of dots of water on both sides. It's easier for you to figure out. I'm slightly confused. Alright? Red is going to represent our solute. Alright?
Oh yeah.
Make the blue dot.
Albert, no.
No, she wants you to leave.
Alright. So, right now, you can already see where there's more solute, right? But let's think about it this way. So right now, there is a 3 to 6 ratio, right? So 50% for concentration, right? That's not a 5. I'm just going to leave it in fractions for a second. Sure, it's easier. Well, I'm actually not going to, so that we can easily compare over here for a second. But
Oh, good.
just saying.
Got
Now over here, I have 6 water, and I have 1, 2, 3, 4, 5, 6, 7, 8 solute, right? The red dots have to stay where they are. So in order to make these two fractions closer, I have to move the water, right? So what's going to happen is some of these water molecules are going to come over here to try and equal those out. Does that make sense as to what's happening?
Yes.
Yeah, so the solute doesn't move the water.
The solute's not going to move. What's going to happen is this guy's going to come over here, this guy's going to come over here, that guy's going to try and come over, so that we end up with a closer situation. Because then that would be 8/9, and
You just want to balance.
isn't that a lot closer?
Yeah.
So it's trying to balance itself out. This, though, is why it never stops. Because it might not ever get to that perfect equal, but we're getting a lot closer, and if we keep just exchanging it, it stays in that happy state. Does that help you kind of visualize it a little bit better? All right. Yes, sir.
Wait, but if you're taking water out of the solution, doesn't it make it less diluted? Like if it's got so much water in it, and you're taking the water out, it's got more solute, so does that make it more concentrated?
Yeah, that's what we did. We went from having a 1/2 over here
Oh.
to a 1. But we also went from something bigger to 1 to something less. That equaling out.
So now that they have like the same like similar ratios, would the actual like whole thing from like the left one move into the right one so they have like the same amount of molecules, or would they just stay with the same ratio instead of like the same amount? You see what I'm saying? Okay, because there's like more molecules on the
Total?
on the left one, but there's more, and then the one on the right. So would they ever? So when the one with the more molecules, would those molecules ever move into the small one to equal it out?
Yes. That's just a much slower process because that requires usually an active transport to move that other stuff. We're not going to do that if we don't have to because it requires energy. That's why we move water when we can. Water moves easily, freely, quickly. So we move the water. Yes, sir.
2 questions. So from here it would move to the right.
A little bit, yeah.
Okay, and second question is, So a dissolved substance needs active transport to move over?
Some of them. It depends on the substance. And it's not necessarily dissolved, but yeah.
So can, like, the blue and red move over together? If the blue
At
and
the
red
same
move over-
time, through the same thing? No, not unless we have like an endocytosis process. Yes, sir.
So for the one on board, why can't it go from A to B?
Because if we did that, it would just be diluting B down that much more. I drew that situation up here. So.
So we're comparing the ratios, not the total.
On each side you're doing.
Because concentration is a ratio,
Okay.
right? Olivia?
1 second. Okay, so like what? It's like how it said glucose and fructose. What if it was like something even bigger versus?
Give me 1 slide.
Oh, okay.
Yeah, that's
Next
actually
week today.
Can water ever diffuse in itself? Elizabeth, is this lined up? Like, can you see the board?
There's water on both sides. Like you do that.
You're
Like,
completely on the
it's-
whiteboard.
there
Oh,
were
yeah.
no red dots. There were only blue dots. It's just very simple.
Yeah, it would just be simple. Alright,
Can't see it.
so here, now I have 100 molecules of glucose and 100 molecules of sodium chloride. Now what's going to happen?
Oh, I know.
It's gonna go from.
It's gonna go to B to A.
Oh,
Now here's where last unit comes
B to
in.
A, B to A.
A to B.
B to A.
What kind of a molecule, chemically, is glucose?
It's a sugar
Monosaccharide.
polysaccharides. Wait, mono.
Bigger picture.
Polymer. Monomer.
It's covalent.
That was like 2 years ago.
No, we only had one test.
That's gross.
You only had one test.
Alright,
I only-
what type of a molecule, or compound actually, is sodium chloride? Okay. Covalent compound, glucose in particular, is it polar or nonpolar?
Non-polar.
It's nonpolar. Do nonpolar solutions or compounds dissolve in water?
No. Wait.
No. So, compare that to sodium chloride. It's ionic. Do ionic compounds dissolve in water?
Yeah.
When it dissolves in water, that means it separates into its parts, right? So glucose doesn't separate. So I still have 100 things in A. Sodium chloride dissolves in water. So now I have 200 things because the sodium and the chloride split up. Sodium chloride dissolves in water.
100 of
It
sodium.
went into
So
it
yeah,
changed
you get
into sodium
100
and
sodium
chlorine.
ions and 100 chlorine ions. So now I actually have 200 molecules, right?
So it's A to B.
So I would actually move from A to B. Yes, ma'am.
But that doesn't make sense since glucose is a lot bigger.
Doesn't matter what size. So that's why I said, Olivia, hold on for a second.
Size doesn't matter, Stan.
When we're talking about the osmosis, it's not size, it's number. And that's because when we talk about concentration, it's all just a ratio of how many, not what. Now the what could play a factor in this sense because does it dissolve or not, but not the size. So that's why I said give me a second. But.
So as long as it's not covalent versus ionic.
So you have to ask yourself, is it covalent or is it ionic? And if it's covalent, is it polar or nonpolar? And this is where your understanding of your macromolecules comes in, right? Because our macromolecules told us which ones were which. Our carbohydrates are all what? Nonpolar. All right? Our lipids,
Non-polar.
Polar.
which one are they?
Non-polar.
Because they don't dissolve in water.
Because
But they don't dissolve in water because they're
Hydrophobic.
hydrophobic.
Wait, so they are polar?
So no. They're non-polar, but they're hydrophobic, so they're not going to dissolve in water. So this is where this was the other question from third period, which is there going to be old unit information on this test?
Yes.
Yes, but more in this sense, right? You have to be able to process stuff from old units in order to answer our new unit questions. That's how the AP classes work, is we continually build on our knowledge. So it's not like I'm going to go back and give you a straight up essay on the carbohydrates versus the proteins. That's not how. But now I could do, and I'll say it on this one, all right? Facilitated diffusion requires our transport proteins. So now I could go back and give you a scenario that says, okay, what happens if I change my amino acid sequence of a carrier protein? Now you have to understand, okay, amino acids come together to make polypeptides. Polypeptides fold on themselves based on R groups to make our proteins. The proteins determine the function. Well, if I change the folding, I change the shape. If I change the shape, I change the function. If I change the function of a channel or a carrier protein, now it may not accurately transport the right thing across the cell. So now we're taking it that step further, right? So there's our big picture we're trying to aim at. Can we take our little details and can we go step 1 to step 2 to step 3 to step 4? That kind of makes sense?
Mm-hmm. Wait, sorry, back to the last thing. Can you explain why it goes from smaller to bigger? Like this less concentration than more?
Setting.
Okay,
We're trying to even out because concentration is our ratio.
Yeah.
So a smaller ratio means like I'd have 2/5 over here and 4/5 over here. I'm trying to equal them out, so I'm going to move something to make them be 3/5 or make them closer, right? So I'm going to move water to make that happen.
So is it always smaller to bigger, or is it
Smaller concentration of solute
Yeah.
to bigger if we're talking about water.
okay?
Yes.
Got it.
Yes, ma'am.
Okay, with the last slide, about
Leave our hair alone.
about the NaCl and the glucose,
Mm-hmm.
why doesn't the glucose break?
Because it's non-polar, which means it doesn't dissolve in water. Yes, ma'am.
So nonpolar and polar like work the same way as covalent and ionic?
So in covalent, we have our polar and our non-polar, right?
Mhm.
The stuff that can dissolve in water is polar covalent and ionic. Non-polar is the only thing that won't dissolve.
So what if you had like two non-polar molecules that were different sizes?
Different size wouldn't matter. Because, I mean, like the first one we looked at, fructose and glucose, they're different sizes. It didn't matter. Alright? So of our transport proteins that we use in facilitated diffusion, we have 2 types. We have our channel, which allows a corridor for specific things to go through. So like water. Water uses a channel protein. What's the specific one for water?
Aquaporin.
The aquaporin, all right? So this is good because think about it in terms of water. We want to be able to move high quantities in and out quickly when necessary. So we want this open pathway. As compared to, give me a second because I want to show you this and then I'll come back. A carrier protein, which picks up one at a time. And it's like a, think about it as the difference between an open door versus the rotating doors, right? Like what's the you know the ones that you walk in and you can fit like one person in each slot? That would be your carrier protein. It slows down the traffic a lot versus if you just had an open walkway, a whole bunch of people could pass through at the same time, right? That's the difference. The carrier protein is like your revolving door. Your channel is like your open passage. Right? That is what you need to know through for the test.